Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of UW-Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
So we are really excited to have Dr. Cecilia Lee, who is an associate professor in the Department of Ophthalmology and is the Klorfine Family Endowed Chair at the University of Washington. Dr. Lee is a medical retina specialist and a clinical epidemiologist and serves as the Director of Clinical Research in the Department of Ophthalmology. She also currently co-leads computational ophthalmology at the University of Washington, where her research focus is on leveraging big data, machine learning, and data science to evaluate clinically impactful and meaningful questions in medicine. Additionally, Dr. Lee serves as the program director of the recent large NIH initiative called Bridge to AI, designed to collect multimodal data from 4,000 diverse participants in various stages of diabetes to allow AI or ML approaches to better understand the development and progression of diabetes. Today, she's going to be talking on basic concepts of big data and how different sources of large data sets can be combined. And her talk title is Big Data Applications in Ophthalmology. Dr. Lee, over to you. We're really excited to have you and really excited to hear about your work.
Thank you so much for the kind intro. So I am going to share my screen. So I am so honored to be part of this data learning series, and hopefully I can kind of really give a brief intro in terms of what we mean by big data, and then how we can apply not only just in ophthalmology. Hopefully I can show you the breadth of big data application across ophthalmology, and then also in between other subspecialties in medicine. So I have no disclosures. So I'm going to talk about just the general concept of the applications of big data in ophthalmology. I'm going to share some of the examples that we have done to connect the eyes and the brain, and then finish with the next steps and future direction, and introduce our currently ongoing study. So what is big data? So simple way to think about it, or a simple way that I think about it, is if there are data sets that are just too large to open on a traditional spreadsheet, then it's probably a big data. Of course, the complexity and there's a huge range in terms of the size and the complexity of the data sets. You might have heard of big threes or big 3 Vs or 4 Vs to describe what big data are in terms of volume, velocity, variety, and veracity. But it's really, it can vary quite a bit in terms of the amount of data sets or amount of the data fields that it may exist, and then also the breadth of the complexity or the, yes, of the data sets. So why are we interested? We are ophthalmologists. I'm a medical retina specialist by training. And I think it's because, first of all, we are entering this new era of passively accrued large data set era, meaning when you go to see your doctor or when we see our patients, all this clinical data are stored in electronic medical data. And not only that, all this imaging data are huge, they generate huge data files, and they're just passively being collected. And not only that, eyes are really unique in the sense that eyes are really one of the organs that is most prolific in terms of data collection, so that we have more people than ever in terms of getting eye data, and then more data than ever in terms of getting eye data from each individual. So this is an example of a person coming in and getting this non-invasive imaging that can be done within a few seconds. And then this is an image of the fundus photo. And this is an optic nerve, and then this is the retina, and then these are large vessels. And this is an abnormal fundus photo, typical of bad diabetic retinopathy. So somebody that has diabetes, and you develop all this like bleeding, all this exudates. And this is a machine learning. It's a work of one of our faculty member at UA who has developed model that can quantify and segment all these abnormalities that exist. This is a work of Ricky Wang from our department. This is called the optical coherence tomography angiography. And this is all non-invasive without any dye. You can actually visualize even capillaries at the entire retina, which is remarkable. So prior to this imaging modality, we used to inject dye, or we used to just kind of use the fundus photo to delineate large vessels. But imagine how much sensitive data we can gather by being able to quantify all these tiny little capillaries and then abnormalities that come with that. This is an image called optoretinogram. This is a work of Ram's Vision from our lab. And he is able to stimulate one single photon, so one single cell that exists in the retina that is responsible for vision, and detect its response, which is remarkable. So we can go as wide as we want and then as deep as we want in the retina. So it does create a very multimodal data set. So again, this concept of routine eye clinic visit today, we get imaging, we get all this clinical, and then we can really combine the imaging database with the electronic records, and then this all get ingested into big data pipeline. So in addition to the electronic medical records data, what are some examples? So we can think about census, CMS billing data. But then we can go as far as Earth data, OpenStreetMaps, or even social media such as Twitter. So why are we going beyond ophthalmology, beyond medicine, and then all the data sets that are really surrounding us every day? It's because we think that when we are combining these various sources, essentially, we are able to gather novel insights and novel discovery that allow us to predict early who might develop a disease or who might not respond as well to a certain therapy. We become better at diagnosing them. And then also, hopefully, we can optimize treatment that is specific to that individual because of all these various data sets that provide us different risk profile that is really targeted to that single individual. So that is the kind of big picture. And I'm sorry, can you guys see this? A participant has enabled. Just want to make sure. Are the slides okay?
Yeah, the slide.
Okay, great. Sorry, I think I'm just seeing another text. Okay, so I'm going to start talking a little bit about the IRIS registry. So this IRIS registry is called Intelligent Research Insight. It's sponsored by American Academy of Ophthalmology, our national organization in terms of all the ophthalmologists and then some of the optometrists as well. So this is actually the largest clinical data set that exists in the world. It started in 2014, and now we have over 70 million unique patients data, over 412 million patient visit data, so unique encounters. And then this is coming from more than 16,000 eye care providers across the United States. The University of Washington is one of 5 sites in the nation that has research access to this large IRIS registry data set. So this is quite huge. So we wanted to really leverage this data set to really get new insights that it would never be possible without this type of big data sets. Before I kind of get into the details of our study, I wanted to briefly introduce you to this disease called age-related macular degeneration, or AMD. So this is a normal fundus photo of somebody. So this circular area is the optic nerve. This is the fovea, which is the center of the macula. Macula is here, which is the center of the retina. And these are large vessels. Now, this is the middle photo is an example of dry AMD. And you can see this kind of yellowish spots. Those are all deposits. And you can imagine that if you have all these deposits clumped with either below the retina or within the retina, the light can't really penetrate and things are going to be really blurry. And this is a typical image of a wet AMD where you have big bleed. And obviously, you know, if you have a bleed, a large pool of blood within the retina or underneath the retina, then the light is not going to penetrate and you can't see. This is unfortunately the most common cause of blindness above the age of 55. So what does that mean for our patients? And you might know somebody or who has been impacted by this AMD. This is kind of what you see. It unfortunately affects the really center of your vision. And so actually things are pretty clear in the periphery, but when you're actually trying to look at your grandchildren or your spouse, you just can't see the space. You can't see their faces. So it's a big problem. And then we've been trying to really study, you know, for decades, this really huge problem. So then now we wanted to ask, well, can we leverage this big data to obtain novel insights in AMD risk? Because we know some genetics, some genetics, but we just don't know much beyond that. And then we can't really know what drives somebody to develop from dry AMD to this big bleeding problem to wet AMD, or, you know, we can't really know whether there are any factors that we could address for prevention-wise or decreasing its progression. So this is work in collaboration with Emily Chu, who's at National Eye Institute, and Mike Mnookin from our department, and then Matthew Hunt, who was a medical student who took a year off with us, and then now he's in Iowa during ophthalmology residency. And we wanted to look at some environmental risk. And, you know, this concept of the environmental risk factors that impact, you know, disease development or progression, it's not a new idea, right? We always think about, oh, is the air pollution really bad for this and XYZ, but it has not been really possible until nowadays that we can combine all these large data sets. First, ingest large environmental data, and then also has a matching data set that is large enough to get those kind of insights. So what do we do? So using the IRIS registry that I just mentioned, we extracted all the data within the IRIS registry that were seen, patients that were seen in 2016 to 2018. We included only patients whose provider location was available. And we extracted age, sex, race, insurance, smoking, and phakic status, meaning whether they had a cataract surgery or not, because we believe that that does have a lot of impact in terms of course, exposure to the environmental factors and other risk factors. So out of all those patients, we divided them into no AMD group. So this is a normal fundus photo, into dry AMD group, and then into this wet AMD group. And then this, so there were two types of wet AMD group, meaning wet AMD group that is actively getting treatment. And we usually treat them with the injections. So we were pretty confident that they were actively getting treated because we had access to their CPT code. And if we didn't find a CPT code, or if their diagnosis ICD code did not specify active, then we kept them as an inactive wet AMD category. Before I kind of share more of what data we have extracted, I want to kind of give some background of ZCTA. I think many of you might already be familiar. So the zip code tabulation area is, I think of it as a zip code for environmental factors. So the reason why we wanted to get ZCTA is because US Census data has determined, identified all the latitudes and longitude that are specific to each ZCTA internal points. So then it kind of gave us the point where we can match to the nearest location of each environmental measure. So you'll see that key in doing big data work is that you kind of understanding the setup and the limitations of each data sets, and then you do have to make some assumptions, but then also later on, you do have to recognize those limitations and assumptions so that you understand its generalizability. But in terms of the environmental factors that we extracted were elevation, and NOAA, the National Oceanic and Atmospheric Administration data, has 12 measures average over 30 years. And then also the National Renewable Energy Laboratory, or NREL, data had global horizontal irradiance or direct normal irradiance, so GHI and DNI data. So I'll show you why we are interested in GHI and DNI, because that has to do with some sunlight and sun exposure. So DNI, the direct normal irradiance, is the direct sunlight that are being exposed on the surface. So they have this collecting surface that is orthogonal to incident rays, so they can measure what is the amount of direct sunlight that is being provided at that time. The GHI, though, it's kind of everything. So they measure the direct sunlight, but then also kind of diffuse. So global horizontal irradiance, it's kind of the diffuse sunlight. So I'll just, and then this will become clear why I wanted to make a big point out of DNI and GHI. In terms of the analysis that we did was we wanted to do mixed effects for logistic regression model for predicting whether they have any AMD compared to versus no AMD, or the active wet AMD versus all other AMD, or dry AMD versus any wet AMD. And we chose those kind of three models because those are very meaningful clinical outcomes. So you care whether they develop any AMD, and you also care if you develop wet AMD that needs treatment, and then you care about whether there is a discrete differentiation that we need to do between dry and wet AMD in general, regardless of their treatment status. So that's kind of how we made the outcomes. And this were going to be a huge compilation of all the environmental variables. So we wanted to do bound barony corrections for all of the models. So we had near 52 million people's data. And when we divided into those 4 groups, so we had about 48 million with no AMD. Wet AMD, we had about 2.6. That's, I'm sorry, dry AMD, so that's non-exudative AMD, that's dry AMD, 2.6. And then inactive wet AMD was about 389,000. And then the active wet AMD was over 500,000. And then we kind of talked about how they had to be older, and then also we needed provider level postal code data. And we also included, only included people who had access to the retina care provider who would provide the retinal, the injections for the wet AMD. So then with the inclusion criteria applied, we ended up with 8.6 million, and then about near 1 million for dry AMD, and then about 400,000 total in the wet AMD kind. So now I'm going to move on to large forest plot. Just to orient you, so this is for the outcome of kind of knowing any AMD versus no AMD. And this is, forest plot of odds ratio of 1. So the dotted line means that it's odds ratio of 1, meaning that there's no association. And then I'll show the red dots as significant. So we didn't really find anything that were really significant comparing any AMD versus no AMD category. And so was here in the dry AMD versus wet AMD. But when we really looked at who are the people who require wet AMD or develop wet AMD that requires treatment, we saw a huge signal of significance in terms of GHI. That was a global solar irradiance as a risk factor for wet AMD. But then DNI, that was a direct sunlight as being protective or protective association against the wet AMD. Which is a little confusing, but we'll kind of go through that. So then again, GHI and DNI. And then before we got excited about these, we wanted to look at other data, other risk factors that we already know that they are known to be either risk or protective, because then that kind of gives us more reassurance or that we are on the right track in terms, and then we can trust our results. So this is a really busy slide, but I want to show that when we were just comparing the demographic factors, yes, we know that Black or African American ancestry is a well-known factor to be protective, or the white Caucasian ancestry, European ancestry is heavily associated with the development of AMD. So we kind of saw that effect. And then if you're a smoker, then that is definitely puts you into risk profile. So we actually saw that in all three models. So that gives a reassurance that our data sets does show the results that we would have anticipated. And then pseudophakia, meaning that they had cataract surgery, then it is shown to be more to the right. Do you have a risk associated with AMD, but that could also be confounded by age, and then there might be additional confounding by that. So at that time, when we added all these risk factors, including demographics, we still saw a significant association of GHI towards the development of active wet AMD, and then DNI as protective association of wet AMD development. One thing that everybody asks, or even we ask, oh, is that just a function of latitude? Because if you live in a higher altitude or higher latitude, is that really what's driving the solar exposure? And interestingly enough, latitude does have association, but really didn't have. It was not, comparatively, it was very weak association. So we were kind of scratching our head and trying to explain why that could have happened. So to summarize the novel findings that we found was that if you have a lot of direct sunlight exposure, then you develop less of wet AMD. But if you have a lot of exposure to diffuse solar radiation, then you develop more wet AMD. And we thought that, and then that really was not impacted by the latitude. And we thought that maybe the reason is because sunlight is probably negatively impacting to the development of wet AMD. But when you have a lot of direct sunlight, let's say that at like a noon, or whenever there's like a high direct sunlight, then maybe people are likely to just stay indoors or wear sunglasses. So you have less exposure to overall sun. And then you might not mind if there's more kind of diffuse sunlight, like in Seattle area, when there is not that much, direct sunlight, but then there's a lot more diffuse sunlight, and then maybe we will be more, those people would be at a higher risk of developing wet AMD. So big data are meant to generate new hypotheses that we have never even thought about, and then actually this results were quite surprising to us. So obviously there are many limitations. So we're not dealing with the lifetime information on each individuals. And then we don't know how different it would be because the solar irradiance measure that we got is from environment. And then that does not mean the same to ocular exposure. For instance, they could have worn a lot of sunglasses, so we don't really know. And then this is, even though this is the largest registry that exists in the world, it's confined to the registry population, and it's retrospective. So we were able to publish this paper. And then there are lots of papers that we were able to publish just using that large registry data. So I'm going to kind of move towards our second portion of the talk about using big data. And I really think that one of the biggest strengths of using big data in research is for making novel connections and then developing new insights and coming up with new directions of research and then healthcare. I'm going to share the example of how we were able to connect the dots between the eyes and the brain. Currently, almost 50 million people live with Alzheimer's or other forms of dementia in the world. And from Lancet Commission report, this number is going to reach more than 153 million by 2050. So it's an alarming number. And if that is not alarming enough, then every 65 seconds, someone in the US will develop Alzheimer's disease. So it's a really serious disease that impacts all of us. I'm sure you are, somebody, and then maybe some of you have been impacted by a family member who knows with Alzheimer's or other forms of dementia. So why is it so hard to study Alzheimer's disease? It's because we don't quite understand the mechanism. So when you actually don't know how it develops, it's hard to treat them. And then there's such a range of, and then, Alzheimer's disease is just not one disease. And then not only that, we don't have a definitive method of diagnosing them. It's not until they are actually dead, they're deceased, and then we actually look into their brain, we can definitively say, oh, this person had Alzheimer's disease or not. And we don't have a scalable biomarker of early stage of the disease, unfortunately. So even people that we are recruiting them into clinical trials for new therapy, their brain might not look healthy, and then it just might be too much atrophied before we can recognize the symptoms or signs, or we can diagnose them so that they can be put into trials. And our lab is leveraging on the fact that the eye actually has developed from the brain. So during our embryology, the eye actually develops as part of the brain, as an outpouching of the brain. So this is the outpouching of the forebrain that gives rise to the eye here. And then so this is kind of the retina, which is the neural tissue that is part of the CNS, so that it is true that retina and brain share the same neural tissue. And then so it is not, there is truth to that when you say that the eye is a window to the brain, or the eye is window to the soul or the rest of the body. So we are really using the eye as a window to the brain as a potential modality that can provide us with early, scalable, non-invasive biomarker of early stage of Alzheimer's disease. So I'm going to give you two examples of our projects. I do need to provide some background of the adult changes in thought study. So we initially started this collaboration with Paul Crane from GIM Department of Medicine at University of Washington, and Eric Larson, who used to be at Kaiser Permanente in Washington, and now he's at UW. So this cohort actually started in 1994. It started with people who were 65 or older. They were recruited from Kaiser Permanente Washington membership role before it used to be group health. And they had to be cognitively normal at the time of recruitment. But every 2 years, they went through a battery of cognitive tests. And then if they went below the threshold, then they were they received a multiple battery of cognitive testing, physical exam, and they were diagnosed with Alzheimer's or other types of dementia with the research criteria diagnosis, meaning that this was done in a consensus panel with multiple experts who decided or agreed upon the diagnosis of Alzheimer's disease, which is really the top quality diagnosis that you can hope for these days. And so far, because they keep a 2,000 maintenance cohort of 2,000 participants who are at risk of developing dementia at all time. So when you develop dementia, then you're out of the study. So whenever there are people who are deceased or they develop dementia, they replenish that maintenance cohort. So that's why we've had over 5,500 who have contributed their time to this study. But because we received a larger funding mechanism now, that they are going from 2,000 people's maintenance course to 3,000. So it's going to be huge. And then we have more than 1,000 brains now that have been archived and autopsied. Because about 30% of the people who are agreeing to donate their brains as well. So again, to just kind of summarize, they are prospectively collected since 1994, and they are all dementia-free, and they are followed at a regular interval. And then, when they are deceased, we even get their brains. But one of the beautiful part of this study is that they were all part of Kaiser Permanente Washington members. And because that is a large HMO organization, they provide comprehensive medical care to all the members. So then we kind of got all this data for free. So we know all the laboratory data, their imaging, their medication, even field area data, and then all the clinical data that have been obtained either in paper charts or electronic medical records and surgery, including all the eye data. So that's really what allowed us to look at this eye data retrospectively without doing much work. I felt like I was, stealing stuff. So we have retrospectively collected eye data from EMR, and then we were really able to gather that. So our first question that we asked was, are there retinal degenerative diseases that are associated with a higher risk of developing Alzheimer's disease? So we chose age-related macular degeneration, the AMD disease that I talked about earlier, glaucoma, and diabetic retinopathy. So these are 3 conditions, sample photos of those conditions. And I'm just going to kind of go briefly that we use about 3,900 participants data, over 31,000 person-year data, and about 800 Alzheimer's disease incident cases in this analytic data set. And again, this is a forest plot that summarizes that finding. So hazard ratio of 1, this dotted line. So age, we know that that is the most well-known risk factor. So that kind of pushes us to the right of the hazard of the forest plot. So it is associated with risk. And education, we know that is protective. Not huge, but there is a protection against Alzheimer's disease. And ApoE4, it's lipoprotein, and E4 allele is the most well-known, strongest genetic risk factors for late onset Alzheimer's disease. So it did have association, a huge association, about 1.6 in our cohort, associated with the risk of developing Alzheimer's disease. And we kind of expected that, so that was good that we saw. One thing that we did not realize, though, was that if your eye looked like this again, AMD, your risk was as high, and diabetic retinopathy and glaucoma. So all 3 conditions were associated with a huge risk of developing Alzheimer's disease. But what was even more remarkable was that they were in line, comparable to you having ApoE4 allele. So if if I saw your eye in clinic today and then you have this AMD picture, I could have told you that, oh, your risk of developing Alzheimer's disease is as a high, or it's the same as me just taking the blood test and then checking for that most well-known risk factor, genetic risk factor. So we published that in our paper, but this really gave us foundation that there's a deep connection between the retina and the brain in terms of understanding potential mechanistic connections between the eye diseases and Alzheimer, so that it just gave us the foundation to go after understanding better the eye diseases and eye condition for Alzheimer's disease research. So our second question was, now that we have established that certain retinal diseases are associated with higher risk of developing Alzheimer's disease, we were interested in, well, if we are to intervene and then make the eye disease or eye condition better, does that have any impact in development of Alzheimer's disease and dementia? Because one thing is finding a new risk factor, which is great, but then it's even better if we can find a risk factor that we can modify, or it's modifiable. So we asked, is there an intervention that matters or that may have a different association in the future risk of developing Alzheimer's disease? So we chose cataract because cataract is the most well-known aging condition of the eye. So it's not even a disease. If anybody lives long enough, then they will need cataract surgery. So our question was, is cataract surgery associated with lower risk of developing Alzheimer's disease? And we now, looked at more than 5,500 people, but I'm not going to go into much details, but I'm going to say that we did Cox survival analysis with the surgery, so cataract surgery, as a time-varying exposure in our analysis. So this is a forest plot. On the top panel, that is the outcome of all-cause dementia, and the bottom panel is the outcome of Alzheimer's disease dementia. So if you gonna go over again the dotted plot, it's the hazard rate of 1. So anything to the right is associated with the risk, and anything to the left is associated with the protective effect. And you'll see that I had mentioned that if you have a ApoE4 allele, then that is the highest well-known genetic risk factor against Alzheimer's disease. So if you don't have that allele, then it's protective. So we kind of saw that in both models of predicting dementia or Alzheimer's disease. But what we didn't anticipate was how much impact cataract surgery was associated with future risk of developing dementia. So it was way onto the left side, and it was actually about 30% reduction. And not only that, when we looked at the timeline, we saw a significant difference between people who had cataract surgery and who did not, even after a decade following cataract surgery. So this was huge. But sometimes, you get nervous when your results sound too good or too strong to be true. So then we went through multiple secondary analysis to kind of prove ourselves wrong, and we just kept seeing the same results over and over. And one definitive sensitivity analysis that we did was really comparing to the impact of another intraocular surgery that does not improve visual function. So glaucoma usually increases the pressure in the eye, and then you get this visual field loss as the time goes, and you get glaucoma surgery to prevent progression of glaucoma. So actually, that's one of the difficult surgeries to kind of talk patients into, because we'll say that it won't get any better. We're just trying to stop it. But sometimes we have to do. one of the things that we were struggling was that cataract surgery is a optional surgery and it's an outpatient surgery. So they need to have at least some kind of good health to be able to come into the clinic and to be able to tolerate outpatient surgery that you get in the, you get the surgery and then you go home that day and then you follow with your eye doctor the next day. And glaucoma surgery is the same. It's not an optional surgery, but it's an outpatient surgery. So we thought that, well, why don't we compare the impact of cataract surgery to glaucoma surgery? And because they're both intraocular outpatient surgery, but only cataract surgery improves visual function afterwards. And we saw that primary model, so the association between cataract surgery and the risk of developing dementia was 0.71, so about 30% reduction. But when we looked at glaucoma, it was pretty null. So that really gave us reassurance that the result that we saw, this protective effect, was really specific to cataract surgery and not the fact that they were healthy enough to undergo an intraocular surgery at an outpatient setting. So what are some potential mechanisms? Well, if your vision was this blurry and then now things are sharper, maybe you're less depressed, you're more likely to exercise or become more social or take on more new hobbies, all of which have been shown to be protective against dementia development. And we kind of think about the better neuronal stimuli, both visual stimuli, and then that translates into better neuronal stimuli in the brain. And then also there's a concept of cognitive overload, meaning that if your vision is too blurry, then your brain is too busy trying to figure that out and then can't really process other memory function well enough. So that could be another reason why. One of the interesting hypothesis that we came up with was that there is specific cells in the retinal ganglion, in the retina. It's called intrinsically photosensitive retinal ganglion cells. And then these are really interesting cells because they are not responsible for vision at all, but they are responsible for circadian rhythm. In some animal models, they have shown to be associated with mood and some cognition even. And so we were thinking, well, that could play a role. But what was really interesting was that these RGC cells are exquisitely sensitive to blue light stimulation. So if you actually shine bright light, just like diffuse bright light, they're not as stimulated. But if you shine the bright blue light, then they get awakened right away. And we know that cataract that's making lens so yellow is really good at blocking blue. So I was surprised that many of my patients who do cataract surgery, who get cataract surgery, the next day they come in, they don't say, oh, my vision is so much better, but sometimes it'll be like, my world is so much bluer now because, that yellow tint has been lifted off and then they just see all this blueness in their world. So we thought that, well, maybe there is a role of intrinsically photosensitive RGCs, but obviously that needs to be validated and further studied. So that's what we published. And then we received a lot of media attention because this is so home to so many people regarding cataract and dementia. So what are we doing with this study? Just to kind of loop back into what we are doing. So right now we have about 350 people who are recruited into IACT, meaning that we are recruiting all the ACT participants into our study, where we do all this visual function study, including all the sophisticated retinal imaging. We're hoping to really generate that type of data set to do more discoveries and then to better understand the connection between the eye and the brain. So what are some next steps and future directions? So I'm going to say that so far I have introduced you this area, this concept of big data, but I haven't really talked about any machine learning AI approaches. But I really do think that it's this marriage between big data and machine learning that allows powerful discovery, because there is more data to mine, but without machine learning approaches, we won't be able to mine all the big data and then train sophisticated models. And then that's the reason why we are getting all this random imaging from IACT study participants. So is that idea far-fetched that we can get all this sophisticated random imaging to develop novel insights to understand our brain, and then even greater health, like stroke, and then cardiovascular disease, kidney disease, heart attacks? So I would say no, because in 2018, you might remember this paper that involved Google and on UKO Biobank. They were, so this is the fundus photo. Now, by now, you are probably familiar with the fundus photo, the retina, but it's remarkable how they were able to train model that can pretty precisely predict the age, sex, even how big your blood pressure was, and whether you're smoking or not, and then your body mass index. I look at people's retina all day long, and there's no way I could predict that. So clearly there are a lot more hidden signals than our human eyes are able to appreciate. So if you are able to get even better data, even more sensitive data than fundus photos, and we are able to really develop models or to extract all the data that clearly exists, hidden latent features, to understand better other diseases, I think that is certainly possible. So NIH kind of recognized this potential with using machine learning AI to understand really better our health, and then that impacts all of us. But remember I had said about this marriage of big data and machine learning. So machine learning can't really excel unless there's large big data sets to train. And then big data really can't survive by itself without sophisticated AI algorithms who can really take advantage of that. But we recognize that there are no large existing data sets that are really good for AI models, because they are either too skewed towards healthy people's data or sick people's data, or they are not diverse enough so that we get into trouble with the model development. So NIH provided this funding announcement about Bridge to AI to create hypothesis-agnostic flagship data sets that would be useful for many AI models in downstream. So we were one of the 4 projects in the nation that were funded for this large efforts. So our project is called AI Ready, or AI Ready and Equitable Atlas for Diabetes Insight. And our goal is to create a multidimensional, ethically sourced data set in diverse people for studying salutogenesis in type 2 diabetes. What do I mean by salutogenesis? So we are very familiar in terms of health researchers, the term pathogenesis, and kind of try to figure out what are the risk factors that make somebody from health to disease. And salutogenesis is the opposite. It's a promotion of health from disease state to health stage. And we were fortunate enough to gather a large army of experts in many different fields, and actually ophthalmology is the minor portion. So across multiple subspecialties of medicine, including ethics, and then 7 different institutions. So how do we plan, or how do we envision ourselves in doing this salutogenesis prediction? So we are borrowing this concept of, we're borrowing the previous success of single-cell RNA-seq technique, that there are some powerful machine learning techniques that were used in single-cell RNA-seq analysis, that you get tons of data points of cells, and then this machine learning will make the data points of the cells that are really close to each other, right next to each other, and then the cells that are very different from each other, very far away from each other. So then they were able to create something called pseudotime manifolds. So because there are so many cells at different time points of the development, if you actually apply that technique, then you can kind of see a timeline or atlas of how a cell from the embryonic stage goes to fully differentiated stage. So you can kind of, even though you're doing analysis at the cross-sectional level, cross-sectional time point, you're able to see kind of the entire history. So we are thinking of the same thing, that if you are able to generate enough of multimodal data that is well-designed and well-balanced, then when you apply this technique called pseudotime analysis, pseudotime manifolds analysis, then you should be able to see how people are lined up in terms of the healthy progression versus disease progression. So what type of data are we recruiting? So we are balancing triple balancing of people between, so we're collecting 4,000 participants, 1,000 normal, 1,000 lifestyle controlled, 1,000 oral medication controlled, and then 1,000 insulin dependent. And we are also balancing between white, Black, Asian Americans, and Hispanic. And then we're balancing male and female there. And this will be from 3 data collection sites from UW, University of Alabama at Birmingham, UCSD. And then we are even working with the Joe and the team at Native Biodata to involve American Indian tribe. So what are we collecting? We're collecting lots of medical history, demographics, vitals, a lot of surveys to include to include social determinants of health, depression, cognition, and I forgot to mention that we're getting a lot of blood work, urine, and EKG, 12-lead EKG, visual function, lots of imaging that I had talked about, activity, heart monitoring, continuous glucose monitoring for 10 days. And then we are even building in-house environmental sensors that will be deployed to each patient's home. And then we are getting whole genome sequencing, and we are banking plasma serum and then cryogenic storage of iPSC cells. And what is even more is that we'll be open sourcing all this data. So our dream is that any AI researcher out anywhere in the world can download their data sets and then can start working. So in terms of our vision is that they come lots of participants in multimodal data collection, and then not only will we able to do just cross-section the analysis of cross-sectional AI models in terms of understanding diabetes severity, cognitive function, any of the clinical outputs that we want, but then also we'll be able to do some predictive AI model analysis by using pseudotime manifolds analysis. So in terms of the future goals of this is that if any researchers want to generate new data from our participants, and we'll just ask them to deposit all the data back into the open repository. And then we're hopeful that American Indians will be able to participate in this study. But we are really hoping that many future research projects will use this data set, including many of people in the audience. And really, hopefully we can accelerate this data science and medicine by being able to share openly and then really generate this type of data sets that are ready for AI studies. So if you want to know more about our study, there's our website. So take point is that big data can lead to new insights in health. And understanding big data may lead to better diagnostics, predictions, therapeutics, and precision medicine. And all this work could not have been possible with amazing large team that are really incredible. I do want to shout out to Dr. Aaron Lee, who has been a great collaborator. If you don't know, then yes, I am married to that guy, so I do need to be nice. But he has been a great collaborator, as many of you know. And also, I want to acknowledge all the team that are at Kaiser Permanente for IAPT. And then we are always looking for new talents. So please get in touch with us if you're interested in learning more and then even working and joining our family. Thank you.